Hi, Mr. Sapora here. Cell cycle notes part 2. Oh yeah, let's get into it. So in this section, we're going to be finishing up talking about the life cycle of a cell. That's what the cell cycle is. So there's a lot of vocab in here, but don't let it overwhelm you. It's not that bad, actually. A cell has a life cycle just like any living organism here on Earth has a life cycle. And the life cycle of living organisms here on Earth, right, regardless of whether you're a human or a scorpion or a cucumber, pretty much the same. We're born, we grow, we develop, we go about living our life, doing it is whatever it is that we do, and then we reproduce and die. It doesn't have to be as morbid as that sounds, but a cell has a similar sort of life cycle. It has its own little story arc that it goes through as well. To keep this, baton of life moving forward through the universe, through our time. So, in order to do that, all cells go through the first stage of their life called interphase. It's actually where they spend most of their life, right? And the first stage of that is what we call the G1 stage. That is where they grow, and there is growth. The next stage is where, right before they reproduce, they are going to duplicate their DNA, synthesize more DNA, make copies of it so that when they do divide up, each new cell gets an equal copy, an exact copy of the original set of DNA. And then they go through the G2 stage where they are preparing to divide, and they're replicating all of their organelles and things like that and getting bigger. And then we'll get into what we talked about, or what we're going to talk about today, mitosis. So the cell lives in interphase, right? Interphase is this big green part up here, and this is where it does most of its cell business like we talked about in that G1 phase. The S phase of interphase is where it replicates, makes an extra set of that DNA. And then the cell gets bigger and starts getting ready for full-on cell division, cell reproduction, if you will. And that is in that G2 phase, that last phase, where it's just enlarging and doubling up on its organelles and things like that. And now the stage is set for what we're going to talk about today, mitosis. So mitosis is a 4-step process that divides the cell's nucleus into genetically identical nuclei, each with its own full set of DNA. So in order for this cell to split into 2 exact copies, the most important thing that it has to make a copy of, an exact copy, is that DNA. And it's already copied up that DNA. All of that DNA has been copied up during the S phase of interphase, but now it needs to divide it evenly into each side of the new cell, making sure that each side has an exact set of that DNA, an exact copy of all of that DNA, so that it can do its cell business, right? You can't leave one little string of DNA behind in this process. So this is what we're going to be talking about. I showed you this table last week. I'm going to have you fill it out this week for your homework assignment. And we're going to be living and talking mostly about this mitosis, the 4 different stages of mitosis down there as well. Okay, so the 4 phases of mitosis, there's whatever you call this, and an acronym for this or whatever, and it goes PMAT, and P stands for prophase, M stands for metaphase, A stands for anaphase, and T stands for telophase. And so these are the 4 different stages. We're going to learn what they are, but an easy way to remember that is PMAT. Yeah, PMAT. Remember that. Okay, so the first step is prophase. This is the longest stage of mitosis, whatever you want to call it. And this is where the DNA condenses into chromosomes. So the DNA has been copied up, and we have in us, right, long strands of DNA inside our nucleus, right? We have 23 pairs of these strands of DNA that we get from our parents. And it's in there doing DNA work, right? Certain sections of it, genes or coding for proteins. Now when the cell decides it needs to divide, it copies up all this DNA. So it splits, and then it recopies itself using the base pair rule, and you get 2 exact copies. And now you got all this DNA floating around in there, right? You have 23 pairs of DNA. So that's 46 strands of DNA. And now you double up on that. So now you have 92 strands of DNA total in there. 92 long shoestrings of DNA inside your nucleus. All these little strings. Well, let's coil it up and let's condense it into these things that we call chromosomes. And chromosomes are just chunks of DNA that are kind of coiled up around proteins and other things, histones. And you can actually kind of see them with a microscope. They're condensed. It's like taking thread and running it around a spool. So the DNA condenses into these chromosomes, and the nuclear envelope, the nuclear membrane, starts to break down. Okay? So the nucleus in there starts to break down, the covering, and you can actually see the DNA. Usually you can't see the DNA. It's covered up by the nucleus. And that's right here, prophase. What I want you to do is draw it. Draw this right here in your notes. And how I would draw it, let's draw it together, kids. Okay, so make a circle for my cell and make a circle for my nucleus right here. So there's my cell and there's my nucleus. And I'm going to draw my DNA in there. And I like to draw chromosomes as little X's. And right now they're just kind of scattered all over. And they're in there. And now the nuclear envelope starts to break down. So then what I do is I'll erase out little sections here of the nucleus because it's starting to break apart. That's a good way to draw it there. You can actually see the DNA in a microscope. You can see it in there. And if we were in class, we would be looking through microscopes at some of these slides like I have here. This is a whitefish. And these are actually, you can see its chromosomes with a microscope. Right now, you can just see this blurred piece of glass that I'm shoving in your face. You're welcome. So this first one here is prophase. Label it. And draw it. Prophase. There you go. Okay. So this is all different stages of mitosis. Mitosis is the process where the DNA divides up evenly and you get two new nucleuses forming inside a cell right before it divides. It's a really important stage. All that genetic material gets sorted out evenly. And the first part of mitosis is prophase. PMAT, what? P stands for prophase. And here's a picture of what it looks like. You can actually see these pieces of DNA. It's not just one big purple clump in the middle. This is where the nucleus is, but you can see the nucleus dissolving, and you can see the DNA condensing into chromosomes in here now. They call DNA when it's loose chromatin. They have all these different vocab words. It's fine, you don't have to memorize them all. But at least you can understand what it is. So it's like a bunch of different shoestrings or spaghetti strings all laying around in the nucleus, and now we're twisting it into discrete bundles and packages. The nuclear envelope starts to break down again, so you can actually see the DNA just floating around in the cell. The next stage, phase of mitosis, whatever you want to call it, is M, PMAT, PM. M stands for metaphase, and it's the second. And this is where the chromosomes line up on the equator down the middle, and these little protein fibers, I call them spindle fibers, attach to the center of each pair of chromosomes. So now, from prophase, the nucleus is gone, and you've got all these chromosomes, and they line up right down the middle. They call this the equator, right down the middle. And you can see all of the DNA as chromosomes lining up down the middle. One way to remember this is metaphase middle. Metaphase middle. PMAT, what? Prophase, metaphase. And what does metaphase look like? Lined up down the middle. Metaphase middle. So this is metaphase. And we're talking about these spindle fibers. These are little clusters of proteins that act kind of like a come-along or a winch, really. They string out and they attach to the middle part of the chromosome. And what they're going to do is, as the DNA starts to divide, they're going to pull the chromosomes apart and evenly divide up the DNA. 50-50. Each cell needs an even copy of the DNA. Can't just randomly grab pieces of DNA because one cell could have 2 copies of what it needs and the other cell could be missing. And then that cell wouldn't function. So you need to line up that DNA, middle metaphase, and then attach those little protein fibers, spindle fibers to it, and then we're ready for the 3rd stage. Well, where's metaphase? Oh, it's right there. And draw it. Draw it like you see here. Like you see here, and label it. Metaphase. PMAT. Prophase. Metaphase. What's the next phase? Well, here's a picture of metaphase. Here's a picture of it. You can see everything lined up down the middle. Whenever you see everything just nice and even right down the middle, that's metaphase. It's the easiest one to spot because it's so distinct. And you can even see the little spindle fibers, right? So there's a cluster of proteins here called centrioles. Don't need to memorize this. And they shoot out all these little protein, kind of like I said, like little come-alongs, connected to a winch. And then they're going to pull it apart evenly to each side of the cell before it divides. So spindle fibers attach to the center of the chromosomes, and the chromosomes line up down the middle equator of the cell. The 3rd stage, PMAT. It's anaphase. Anaphase is the 3rd phase of mitosis. Now, anaphase is where the chromosomes are pulled apart, and the spindle fibers here start to pull towards each side. So they're pulling over to here, and they're pulling over to here, and they're pulling that DNA in half. And so what you see with that is something that generally looks like this. I'll just go whoop, 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 whoop. So those X's are pulled in half, and now you're seeing the DNA get pulled in the direction towards the blue dots, being pulled to each side. They were on the middle, and now they're moving out to the side. So the chromosomes are pulled apart by the shortening, reeling in of those protein spindle fibers. They are shortening, reeling in, pulling these here. And they then separate and begin to move to opposite ends or poles. What gets separated? The DNA. So the DNA is being pulled to each side of the cell. So where it lined up in the middle, now it's being evenly divided. Again, evenly divided. DNA is, we're not shuffling the deck here. Each cell needs an exact copy. So it copies all the DNA, lines it all up in the middle, then divides it out evenly and starts to move it across. Metaphase is where it lines up in the middle. Anaphase is where it moves to opposite ends. It is in motion here during anaphase. Okay, so Anna goes and breaks everybody up. There you go, anaphase. Moving everything to opposite ends of the poles. That is what happens in this process of mitosis. Okay, PMAT, prophase. Again, all the chromosomes the DNA condenses into chromosomes, the nuclear envelope. The nucleus itself starts to dissolve, and you can see the DNA in there. That's prophase. Takes a long time for all of that to happen, so that's a big chunk of time. When you look down into cells that are actively dividing, clusters of them, you'll see all of this happening in different cells. So you look around and you'll see some in prophase, some in metaphase. Most of the time what you see is prophase because things spend a lot of time there. Things line up in the middle, metaphase, really clear. And then anaphase, things start to move. So there's early anaphase, which almost looks like metaphase. Things are just moving apart. And then there's later anaphase when things are way over to each end now, having been pulled apart. So here is a pretty great, awesome, amazing picture of anaphase. Chromosomes are pulled apart by the shortening of the spindle fibers. Separate and begin to move to opposite ends of the poles. Move them apart. Oh, how about that? Last one. PMAT. Meh. Now we're putting the T in PMAT. Let's do it. Telophase is the 4th and final phase of mitosis. Okay, so telophase is one where the chromosomes begin to uncoil and form chromatin, and you start to see a new nucleus develop. So it is the opposite of prophase, but now you've got 2. It's like prophase in reverse, but now you've got two. So anaphase is everything moving apart. Hopefully you drew that. And now what you have here is the sound. The DNA is starting to uncoil around in here now. Starting to turn more into just DNA. And you're starting to see a nucleus develop. So it's similar to prophase. But you have it twice. One on each side. But it's still one cell. We're still one cell. So you see a cell with two nucleuses. Looks like a googly-eyed alien or something. And you want to draw that. You want to draw that telophase. Right here. Draw it. So draw something that looks like this and label it. Label it what? Telophase. And now, kids, we have PMAT. What? It up. So now, starting at the top, you got prophase. The DNA starts to condense and turn into chromosomes or coil into chromosomes. All that thread winds up into these things called chromosomes, clumps of DNA. And the nucleus starts to dissolve, and you can see the DNA. Cool. Then it lines up in the middle, metaphase. And then it begins to divide apart, anaphase. And then it gets to its ends, and it starts to coil or uncoil, and a nucleus starts to grow, and now you've got telophase. And now you're almost done with the cell division. But now you've completed mitosis, because mitosis is from one nucleus to two nucleuses, where the DNA divides up. And it is an orderly process. It's identifiable. You can see it. You can see it in cells that are growing and dividing. And here is what telophase looks like. Chromosomes begin to uncoil and form chromatin, so no longer really super condensed. You start to see them kind of spin out. And your neatly wrapped thread around a spool starts to just unthread and wind out. And then you start to see new nucleuses, new nuclear membranes forming. You'll start to see this come down the middle too, because now the cell is going to divide in half, and that is the last step. So mitosis has 4 phases. PMAT, prophase, right here. Prophase, metaphase, right down the middle. Metaphase. Anaphase, where everything starts to spread apart. And then lastly, telophase, out here, because you got 2 different sides. Okay, so once again, prophase, metaphase down the middle, anaphase, whoop, telophase. 2 sides, telophase. What's up? And the final stage in the cell cycle, not a part of mitosis, right over in here, cytokinesis. This is where the entire cell splits in half, and you get 2 new cells, what we call daughter cells. So this is telophase right here. Because see, on the outside, you got that one single circle surrounding it. Well, you put a line down the middle, and it starts to divide up even more. Looks like a butt. And then snaps in half, and now you've got 2. No longer telophase, 2 cells. Now you've got cytokinesis. Cyto meaning cell, and to kinese means to divide or split, cut. And so now you've cut those in half, and now you got 2 new cells. Everybody watching this video, every living thing started out as a single cell. And then those cells divide and divide and divide. You started out as 1 cell, you became 2 cells. These cells, then each of these went through their own life cycle, went through interphase, went through G1 where they just grew and developed and did their cell stuff. And then DNA replicated inside of them, the synthesis phase. And then each of these cells got bigger and bigger. That's the G2. And then they started through mitosis themselves. And so each of these nucleuses divided and went through prophase, where everything was kind of clustery in here. So now look at that prophase in each of them. And then each of these went through metaphase, dividing down the middle right there. And then that DNA moved to each side. And now we're into anaphase all the way over to telophase, because now we're going to draw 2 nucleuses around this. One here, one here, one here, one here. Now we're in telophase again. And if we divide these cells, cytokinesis, Now we've got 4 cells. You went from 1 to 2 to 4, and then each of these 4 divided. And if we divide this one up, and this one up, and this one up, so it goes through its own cell cycle, interphase, synthesis, G2, and then starts to go through mitosis again. What's mitosis? PMAT what? So prophase, then they lined up down the middle. Each of these, metaphased, anaphase, they go to each side. And then telophase, each one will get a nucleus that surrounds all of that evenly divided DNA, and then we can divide these again, right? And now you've got 8, and then from 8 you'll get 16, and you keep going and going and going until you get to 37 trillion, which is you and me. Our cells do this all the time, right? And the DNA replicates perfectly, moves across. Little mistakes can add up though, so sometimes it is imperfectly. Those are called mutations. And aging as well. We'll talk more about that at a later date. So, the different stages of the cell cycle. Here's a table, and I put this in the homework sheet. Copy this, fill it out. On your own there, the big stages on the left, interphase, mitosis, and cytokinesis. Interphase is the life of the cell. Mitosis is the division of the nucleus, and cytokinesis is the division of the cell into 2 new cells. It's like you're born, you live, you reproduce, and then you pass along that DNA and you die. Boom. Right? Underneath each of these, we have different ones, except for cytokinesis, cytokinesis. But interphase, there's a G1, an S, and a G2. Mitosis, there's a prophase, a metaphase, an anaphase, and a telophase. PMAT what? Okay, so learn what those different phases are and what they do. This is a part you just need to memorize and understand, but it's a normal, regular cell cycle. When we talk about disease, when we talk about cancer, when we'll talk about other things in this class, when we talk about evolution, we'll be talking about the different stages of the cell cycle because they are important to understand and how they can lead to change over time in a population or, anyway, talk about that later as well, mutations and what those are. Okay, so here we go. You got that table, and I have a review set up, and so we'll do the review actually during a class meeting, our second class meeting of the week. So take notes, fill this out. Here is the note sheet that you are filling out for this one. Draw a label. Knowledge statement, something you learned in that was not covered directly in these notes questions that you learned from watching this video or the video before. And then a question. What is one question you have from this unit? What is something that this makes you wonder about or question? And that can be anything as long as it's related to the actual content. I'm giving points away here, folks. Low-hanging fruit. Go ahead and take it. It's delish. Okay. That's about that, man. We'll get through this unit, and then the next sections we're going to be covering are going to be super fascinating, but we need this information as a jump-off point for that. So learn what these things are, memorize these. We will practice these in class, and then there'll be a quiz also at the end of this that'll cover Cell Cycle Notes 1 and Cell Cycle Notes 2. I hope you've enjoyed this. Hit subscribe, follow me on InstaChat and my Facegram, and watch my TikTok. What about the mustache? You like it? It's pretty good, right? Mustache, scorpion, Sapora out.